Okay, today we get back to the prediction of the effect of mutation on function. And as you all know, the lecture is wrapping up. Examen will be on the January 22nd, on Thursday. We will have a wrap-up meeting, and we will possibly have another wrap-up meeting next Tuesday, and possibly a meeting on something else. Predict the effect of mutations. Ultimately, we want to look at one particular position and predict the effect of that mutation, and ultimately that variation is the background of diversity, if you think about it. These variations are what brings us different sequences. These variations are at the back of the idea of evolution, and evolution is change. By the way, this idea that evolution is change was published in 1858. Some of you may know that the origin of species, or may have seen that from the previous slide, is 59. So the publication of the original idea predates the publication of the book, and it was published by two papers that were read by the secretary of the English Society for Sciences, John Joseph Bennett, the Linnean Society in London, on July 1st in 1858. And they in fact read two papers by two different authors, one being Alfred Russel Wallace, one being Charles Darwin. And that started what we all know as the Darwin-Wallace theory, right? Do we? It's funny, huh? Two guys invented it. One got the credit. One had bad luck and disappeared. So he was the one with the first publication. He was the one who was there first. And this was read only because somebody knew that Darwin was about to write was about to publish his book, and they did not want this one to be the first. That's why they read these two at the same time. So they pushed Darwin onto this plate. I find this interesting. So some things change, I said in this title here, some don't. Now that is something that does change. In this book published in 59, then The Origin of Species, that is the only figure. Do not do that to your papers. Do not do that to your thesis. Figures are important. Put more figures in there. Figures should say everything. So again, we want to say how variants affect the prediction of function, how variants change function. We, on the other hand, know that if we have 100 residues aligned, 33 of them are identical. Essentially, the two proteins have a similar structure. So we are really looking at the little bit of variance that changes a lot. Typically, SNP is defined as a single nuclear polymorphism, and typically is defined not as a single change, but typically, historically, has been defined as a change in the population. So typically, a morphism or a nucleotide change that has been observed in less than 1% of the population has historically not been called SNP. The way we call these things is changing with the advent of sequencing technology. In the past, the reason for that simply was that sequencing was so inaccurate that finding something not often enough, it typically indicated that you made a sequencing mistake rather than you had a piece of data. That's why these words were restricted in the past. Today we can really look at individual variants, and we are good enough at that. And principally the idea is we go from the genotype to the phenotype, so the differences between us, and we can use that, for instance, to look at differences in populations. The migration, we can see that Bushmen differ more amongst each other than an Asian and a European differ in their sequences. We see that there was a migration from Siberia into the New World about 5,000 years ago that is independent of the movement of the Native Americans. We can see that from tiny pieces of DNA, the genetic material. We can define, or Svante Paabo has defined a new species, the Denisovans, from just something found in some cave that was initially thought to be a cave bear. Non-synonymous typically refers to as changing the amino acid composition, and that's what in this lecture we're going to be after. We're going to look at changes of the amino acid sequence. The work started with Jana Bromberg, who is now at Rutgers University, and as I alluded to that, now also an IAS fellow. SNP risk classification, so there are different types of sequence mutations, and missense or non-synonymous SNPs is only one part of it, but they are the most frequent one, and they're in many ways arguably the most important for the diseases we know, at least today, that are related to genetic changes. Let's begin with how we can predict the effect of mutations or variants. The historical first method is SIFT, sorting intolerant from tolerant, going back to work from Stephen Henikoff. I presented Stephen Henikoff before. He's a giant in the field with a very high H index, a lot of high visible publication. One thing that you know from him is Blossom, but he in fact changed biology in many other ways. For instance, he was one of the people who discovered the intron. This is just one thing that happened before Blossom. And many other things. Pauline Nick is the one now at the Genome Institute in Singapore who carved this. Sometimes, I repeat, a number of papers, and this is outdated. The number of papers is not really telling the story. SIFT changed the world, although that is not clearly visible in her record. And the simple idea was, and in many ways we discussed this on last Thursday, we look at an alignment, we compile a Blossom matrix of this alignment, and we ask whether a particular variant is aligned with what I expect from the Blossom matrix, from the diversity that I observe in this alignment, or not. Most simplified, you change a conserved residue, it has an effect. You change a non-conserved residue, it doesn't have an effect. Polyphen is polymorphism phenotyping, the same idea in some sense, goes back to Shamil Sonayev, is now at the Broad, and again for him this is, for somebody as young, this is an impressive number, and in fact it's way behind, and a lot of very visible papers. The polyphen idea begins with the same principal idea, that you want to look at things that are conserved versus not conserved. The difference here with respect to SIFT is the way the math is compiled. So what is a conserved position? So the SIFT idea really is essentially we follow the Blossom matrix, and the probability of a change is more probable to induce something if it is inversely proportional to the Blossom matrix. While now here the assumption is, or the idea behind it is, what we really want to compile is the probability of a particular variant to mess up something in that alignment. That has to do with a variety of factors. It has to do with the Blossom matrix, but it also has to do with what you observe at that position. And it also has to do with how conserved any pair of proteins in this family is. And underneath, polyphen, in fact, is a clever way to recompile the probability of effect given an alignment. That's polyphen, the original polyphen. The next level of polyphen was the same idea, but put machine learning on top of it. SNP S3D is a method that goes back to John Moult. And that idea really is, if for a particular protein you also have you also have the three-dimensional structures, then that tells you a lot about the way you can change that decision. So whether that mutation now is on the surface or inside will make a difference with the way you treat it. Once you have the three-dimensional structure, you can in fact also compile some energy potentials that have to do with proximity of residues in three-dimensional space, and SNP S3D in fact is exactly based on using all this type of information. When Jana started her project, those in fact were the dominant methods that were available, and here's another one. The SDM, site-directed mutator, comes from the Blundell lab, and that in fact goes back to the thermodynamic cycle, and that idea now is some residue mutations simply change the environment of a residue more than others. For instance, the size of a residue. Some of that is visible in Blossom, some of it is not. how much of it is visible in blossom has to do something with the local environment, for which you can somehow compile average potential energy functions. And that is essentially what SDM does. And doing that, it sort of can fit scales in which it predicts the change of binding through variance. By the way, that is Sir Thomas Blundell, a structural biologist who has impacted many aspects of structural biology, including computational biology. So one way, in fact, in which you can gauge whether somebody is important, if there's an important artist making a painting of this somebody. This is the painting here of Thomas Blundell. Another similar idea, pop music, prediction of protein mutant stability changes, comes from Marianne Ruhmann. And here it is in principle a similar idea. The degree to which you affect the function has to do with how you affect the local networks of binding residues. Proteins bind to another protein or another substrate as a group. This group is stabilized by some local interactions that stabilizes, in this particular case, those 5 residues. When I change the degree to which they bind each other, then I will impact function more likely than when I don't do it, or when I do it in a way that disrupt the local rearrangement. And that is exactly what this method tries to predict, the delta G of binding. iMutant, coming from Emilio Capriotti, Piero Fraselli, and Rita Casadillo, predicts the changes in binding energy, has learned from an SVM that uses a database where people measure how the binding energy changes upon mutating in particular residue, some other residue. That's the experimental delta G and the predicted delta G. Is that a good fit? It looks like a remarkably good fit to me. Automute, Josef Weissmann, is another way of doing machine learning, considering the 3D environment. So again, now we're talking about a method that will be applicable only once you have a 3-dimensional structure, or a model of the 3-dimensional structure. And then you can, in fact, again, use through machine learning, you can define what are the local rearrangements that are compatible with that particular environment, right? You look at a residue, you look at its environment, the other residue it binds, and you simply learn to predict changes that within that sphere of what you can reach as a residue matters in order to change, or matters for changing your environment. Yes?
For the method you showed before, for example with the binding style,
yes.
The one before? Yeah, this one.
Pop music?
Yeah.
This is really more...
Do you need to know where the proteins bind to each other?
No. So essentially in this particular case here, what you need to know, but again, here you use statistical potentials that have been used for proteins of known structures. But in principle can get a function here, a prediction for a sequence alone, but typically you would want to have a 3-dimensional structure. You forgot something?
Sorry.
But so it was trained on 3-dimensional structures.
Okay, but you only need to input a sequence.
Yeah.
Okay.
And that is true for all of these, except for this one. That I will not get into. Put up. So I'm showing you, I removed any credentials on this slide. So this slide doesn't come from me. I owe it to somebody else, and I don't want to reveal that person because I'm going to attack that slide. This is a slide for you, because you're supposed to tell me what is wrong with this slide. Let's begin with the simplest thing. Focus on that column. Anything wrong in that column? Anything fishy? This is sort of the sum of counts that possibly helps you, or these. What I'm talking about here is the level of precision that is quoted. You have 1,000 data points, and the table contains two digits resolution. No way in life that with a data set that size here you can support that level of detail. In fact, those are different methods here, and you already see that there's much more coarse-grained difference, and that most likely has something to do also with the data set. So this is absolutely nonsense. That's the first absolutely nonsense in this table. Now, let me give you the background, some history to this. This is a well-known researcher who actually did that slide, and that well-known researcher gave this slide to somebody else who told him, listen, I have 1 minute. In this 1 minute, I will do 40 seconds introduction, and then I can show one piece of data. And that was the answer. Is that giving the answer? Who in this room can in 10 seconds tell me what is on that slide? Nobody. When you prepare data, most of the time what you do is you collect, you sieve through what is right, what is wrong. You do a lot of detailed analysis. One thing that I believe you have felt in the exercises again and again, you ask a question, you find something that could be interesting, follow up, nail it. Don't let it hang in the air. If you really believe something is interesting, follow it through. If you don't believe it's interesting, or when you find out that it was not interesting, drop it. That is something you have to learn. So I had, from several of you, observing you in the exercises, I had several times the feeling that you need more bite. Defined as in you need to deep digger. If you really believe this leads somewhere, go for it. If not, drop it. Make that decision. Make it early on, and then follow. But ultimately, it will lead to the situation where you will follow up in many directions. Leading up to these many directions will create a lot of data, and in the data is a lot of what you learned. Find a way to say it while this door opens. What is the most important finding of your thesis? What is the most important finding of this exercise? What is the most important finding of what you did yesterday? Find a way to say that. That is not the answer. That may be the answer for the appendix. That may be the answer for the detail. That may be the answer if somebody had the time to sit down with you for the detail. But again, at some point you have to approach your supervisor. At some point you have to decide what is the most important item to display for a paper. That will never be the answer. Because nobody's going to parse that in a moment. Okay? That's wrong. I believe since Google started, tables are outdated. Realize that. That's the first thing that is wrong here. But beyond that, learn to focus. Learn to find what makes the difference in what you did. Not for you. I got my degree, okay. But then don't tell me anything other than that. If you believe in the interest of some story, tell the story. And say the most important thing up front. And make it as fast as possible. This never is the answer. That's why, in some sense, it was there. But what it actually says is we have a bunch of different methods that predict the effect, and many of those I showed you before, and that have all some level of accuracy, specificity. They have all strengths and weaknesses. That ultimately is what this slide says. Let's get back to the data. when we did machine learning, so the method, our machine learning method was the first machine learning method out there, and that for the first time got us into the complication of really having to compare different data sets. SIFT, essentially going back to Blossom and use a cutoff, you need to optimize one parameter. For that you need one data set. Very easy to get by. What we need here are data sets that are from very, very different levels of detail. So some data come from delta G of binding. Some data comes from a much higher level. So there's a mutation that changes the way the mouse looks. Important, clear change. But the sort of flavor of those data is very different, right? For the machine learning device, we needed all of that because that was not enough. That was not enough. And that was immediately one problem in our data. It went back to the product mutation database, where we have records such as this one here, unfortunately not maintained. And we got to a data set of 40,000 effect and 14,000 neutral. Again, we briefly discussed that there's a reason why this number is so much lower, because it's much more difficult to get data that something does not have an effect than that something does have an effect. But we know for machine learning, this type of imbalance is a problem. So, how can we get neutral data? No experimentalist is going to give it to you. Find it yourself. How can you?
One could try to somehow simulate it by just doubling the neutral data.
How would you do that? Can you simulate? So again, what we're trying to do, let me recapitulate. We want to have a method that uses machine learning to predict whether a particular variant affects function or not. We have the data set for variants that affect function. We're looking for a large data set of neutral variants. You say simulate it. How can we simulate that? It is true. What I asked for is something that comes from us. And it is true that what we're good at is simulation. And you put these two things together and you have an answer. But the answer doesn't make sense to me. So how would you do a simulation? Go the next step. Anybody?
I would use field alignments.
And then?
And then if one position has more amino acids movement than its neutral position.
But that is exactly what SIFT does.
Yeah.
We don't need to do that. We have SIFT.
Okay.
So that is not going to help us. Yeah?
We can find some protein with same function and we align it, it have different, maybe a lot of mutation, and it should be all neutral.
Everybody understood what you said?
Yeah, but that's the same, right?
No. Nope.
Okay, then I did it.
There's a tiny difference. Well, maybe it's subtle, but it's an important tiny difference. You say we start with an alignment, and that's exactly what SIFT does. He says we start with two proteins for which we know they have the same function. Typically, that's what we want to find in an alignment, but we're not sure if we do. We cannot just take everything that we align and then hope that they all have a similar function. Because in fact, and that goes back to one of the earlier lecture that I gave, the degree to which enzymatic activity is conserved. It's not that very well conserved. But if we took two enzymes for which we experimentally know they have the same enzymatic activity, and if these two enzymes would have some level of sequence similarity, so we would know they have a similar structure and basically they would be essentially the same enzymes, then we would know they do the same thing, they differ at some points. Then we could take the alignment and read off the differences at the alignment and say wherever those two guys differ, they are neutral. Is there a potential mistake in that?
Maybe you have correlated mutations?
Yes, but now you, oops, I go in the wrong direction. Yes. So by the way, that gives me 26,000, and that brings us to a fairly balanced set. You have correlated mutations. Yes, there's one reason. But there's something much more wrong with that. It's true. But there's something more bigger
I would
wrong.
say if you only look for one function, you could miss out others. So maybe you just don't see the effect.
Yes,
because
absolutely.
you don't know how to look at it.
That's true, and that is an important one. But it's even more complicated. So typically what I do, in order to find out that two proteins have an experimental annotation about protein function, I go to SwissProt. In SwissProt I find out all the proteins for which I have the same enzymatic activity EC number for two proteins, right? And then we have correlated mutations, and we have some, they may differ in some other functions. These two are problems, but there's something even bigger than that, which is that most proteins in SwissProt have more than one domain. Mostly, the enzymatic activity comes from one. If it has two domains or many domains, one of these domains is the one responsible for the enzymatic activity. When we do an alignment, we don't know which one that is, right? That is the major problem. Okay? By now, we know, however, that we know two things. Doing this is wrong, and it creates mutual data that is not clean. Not doing it is wronger. It makes it worse. Because we need more neutral data. So going back into the original 40 to 14 imbalance is doing so much damage that we pull enough important, interesting, consistent data here to improve over that. And we know we have to refine this, but we haven't done that yet. When we look at the data in terms of the accessibility, so exposed versus buried, you see that effect versus neutral differs. So the ratio, neutral to non-neutral between buried is 75% and 162%, so that in many ways inverts. And this in some sense already gives you the idea that we may want to treat those two differently. We don't need to know the structure because this effect is predicted from sequence. And that is at the heart of the first method from Jana that uses the SNAP input and gives an output, oops, I'm sorry for the animation, gives an output score between a -100 neutral, +100 very non-neutral. It uses a neural network behind it. And here it is compared for large-scale experiments for which we have a lot of mutations, and for particular proteins, and for these particular proteins you see that SNAP is somehow doing better than other methods. Overall, SNAP performs better than polyphen SIFT, and both of those perform better than random. And there are many, many more numbers that I won't get into. But this is the original slide that I want to get back to, and for that original slide I want to look at something else. Now my first question to you. This uses essentially Blossom, and everybody in this room could have done it by this weekend. Knowing the case, so doing it initially from Pauline and Nick, it was much more than that. Polyphen is way more complicated than that, way more intelligent, way more involved, took a longer time, but in principle that does exist. Going to this one here took Jana 5 years. Okay? Now my question to you, everybody, you as a group, if you did this improvement over 5 years of your time, okay, you get the PhD. But is that something to feel proud of? Is that something that you would feel like, yep, I did it? You have to also put back here. That's where random sits. So that's how much, before you started your PhD project, the improvement was over random, and that's how much you improved over random. It's not that impressive. And if random sat somewhere here, the story would be slightly different. And I would zoom into the difference. But this is not really impressive. Let's look at the slice a little bit differently. Let's just look at everything that SIFT and polyphen predict the same way. And say, well, there are two methods out there that predict the same way a particular effect. Let's call those simple effects. Let's look at the effects that are not simple. And definition of not simple is polyphen and SIFT disagree. For those, polyphen and SIFT perform less than random, but that, is mean. Is simply you pick out everything where they don't work, right? And sure, they don't work for those. That's the definition of your experiment. But now you suddenly see that the method that she created, or that you did after 5 years, in this particular case Jana did, does make a difference. And there are many other ways to explain why SNAP made an important difference. For a lot of our subsequent findings, it was crucial. What I argue for here, sometimes the way you look at what you created with respect to what exists, you have to find different ways of looking at it. And this way of looking at it comes ultimately from observing users. Users, most biologists would not trust a method. They would just run several methods and trust it more. And in some sense, that's what this one does. It says, when these two agree, it may be good. But when they don't agree, go for my method. And that's a very clear recipe. Okay, here are the rock curves, and they are in fact different between buried in red and exposed in blue, and that is for effect mutations. Here, that's for neutral mutations. There is this issue about the reliability index. The reliability index essentially is the strength. So both of those predictions here are predictions for this unit, because this one is higher than this one. This prediction number 1 and prediction number 2 equally are, but prediction number 2, the decision is much less clear. That clarity of a decision you can carve into a reliability index. The reliability index essentially defines 0 means you predict for everything, and for 0 you get a certain level of accuracy. We're around about 80% here, where we have neutral in green and effect mutations in red. So neutral is slightly higher, effect mutation is slightly lower. This is where you are for all the predictions. If you only looked at, say, the 50% of the strongest effect predictions at a reliability of round about 5 and larger, you see that for that subset you get a much higher level of accuracy. So this allows any user, just looking at the output of the method, to predict how good the prediction accuracy at that point is. And that is essentially very, very important. Whenever you develop a prediction method, make something such as this available to users. It's another important message. It's just another case where that is visible. What this one here is, and it takes a little bit longer. Again, here we have effect is predicted strongly, neutral is predicted strongly. We have 3 sets of variants. Very severe in red, very neutral in green, and something intermediate in blue. And you do see that these 3 sets are apart. You do see that there is a tendency, this is a tendency, there is a tendency in this method to have severe effects more on that side, which is what you expect. But in principle, you trained that method only to say effect, not effect. That it captures the severity of a change is by no means put into the system. How do you believe the system learned severity of change? Implicitly. Who told the machine learning device? Obviously the data, but what aspect of the data? Well, we didn't. Because we didn't label the points. This is an after the event experiment. Yeah?
I guess mutations that are very severe also have a higher signal. There must be a really drastic change, and I guess that are the points that the method learns the easiest.
How? Why would it learn that easiest? You're right, you're on the right track. Why? And ultimately the answer is the consistency of the data. So higher difference essentially means more consistency in the signal, so you have a particular projection of a particular point, and points that are somehow similar to it are equally far away. That's the consistency of the data. So clear signal from that part. The more severe gives clearer signals. That ultimately is the way any machine learning device finds a way to separate the points. Data consistency, but you cannot measure. So the important thing again is machine learning versus not machine learning. There is no measure for this consistency of the data. You can compile, if there were a measure for consistency, such as correlation or any other measure you could think about, if that would tell you the truth, you don't need machine learning. You only need machine learning because, short of in fact training your device, you cannot measure the similarity between two input vectors. Cannot measure means you don't have an idea about consistency of the data. You only get that through that prediction device. Okay? Machine learning. Okay. Let's just look at one particular example. So if you know a structure, so this method needs sequence alone. If you knew the structure, then you can take the structure and simply color changes or effect of mutations upon that structure. Now, since we in fact underneath have a method that predicts the effect of a particular variant, you can predict the effect of all 19 non-native changes. Right? And that's shown here too. You somehow can see that some residues are more red and some residues are more blue. The residues that are more red are such that change matters more, any of the 19 non-native. They're most susceptible to change. Now, this overlies the binding site, and you see that one of the binding sites clearly, and if the slide would be better here, you would see the similar reality for the second one. One of the binding sites clearly is where it's reddest. So here's an example where you can simply use the method that predicts the effect upon function of a single variant. You in silico mutate every single residue against every possible non-native amino acid, measure the impact of all that change, color all that as a landscape if you want, and you see a prediction of binding sites popping into your head before you ever know the binding site. Again, we don't use the structure at all for this picture, we only use it to plot the colors, to ground the color plot. And that's an impressive example, and we have seen several of those. And it, in fact, is one of the aspects about SNAP that tells you it has more information than just being able to predict an effect. So 5 years improved by the method that I showed you before. It predicts something that others can't do, but actually it also allows you to do things that others in that sense cannot do. And we got into many years of not really developing the method, which has changed because Max has just worked a long time on the method, but instead to simply milk it, to see what we can learn from the method. One example here is this, experimental mutagenesis. So mutagenesis simply takes a particular residue, mutates it typically historically to alanine, and sees whether or not that has an effect. We can do the same in silico. Right? We can take every single residue, mutate into 19 non-natives, predict the effect, and then look at this change. And then we can see what the average of all 19 non-natives times every single residue is, and see how that average correlates with a mutation of everything against alanine, everything against cysteine, and so forth and so on. So what you observe here is that everything mutated to alanine correlates most with the entire mutation against everything, everything against everything. Again, what the correlation here is with, I take every single position, mutate it to 19 non-native residues, Do that for every single protein residue. Do it for in this particular case, about 200 proteins. And that gives me an average. And now I'm asking, replacing every single residue with alanine, how well does that correlate with the average? 0.9. Replacing every single residue with a cysteine, 0.88. Replacing every single residue with a D, 0.74, and so forth and so on. Let's look at the first two. Is that surprising? Let's look at the first. Alanine. So the highest point here is alanine. The glycine is the second highest point. So we shouldn't have had the line, I take it. So the lines are very misleading. Have to remove them. Don't know whether I still have the data. So imagine there are no lines, and the sorting is alphabetical here. So the second is alanine, glycine. Statements. Does it make sense? The mutation to alanine is predicted to correlate most with a mutation to anything. What do you know about mutation to alanine? What do you know about alanine? Or glycine, yeah?
One could argue that it makes sense because alanine and glycine, they're rather small and don't have any typical. They are not
if all
hydrophobic or polar or charged.
If every amino acid would have a taste, then they would be the least taste, the most tasteless.
Yeah,
of all the
so
amino
they're
acids.
like in the middle of everything. So maybe the average overall is at some point maybe a charged amino acid is not bad, but a hydrophobic one is. And the alanine is kind of in the middle, so maybe always balances out the differences of the different groups of amino acids.
So I believe you would argue that since alanine and glycine are considered the most tasteless, in some sense, amino acids, in fact alanine is by far the most tasteless. That is why most studies are mutations to alanine, because people assume that this is the most neutral thing you can do without introducing a new bias. And this is exactly what this one seems to suggest. Glycine is the next, that is completely compatible with the data. And in some sense you could argue the most surprising finding here might be the cysteine, or the, no, this is serine. It might be the cysteine here. There is an argument why the cysteine is, in fact, ultimately this brings us back to cysteines that form bridges are only some. So most cysteines you can change the few for which the bridge is formed, you cannot, but in the number this is not visible. That most likely explains this. But other than that, in principle, this is nothing exciting, this is nothing impressive. But isn't it amazing? Because the method that we have underneath has learned from some data set for mutations to predict the effect of a variant. It has never learned to do an all against alanine mutation scan. It's amazing that it reproduces something that we know here, right? And it certainly is a different example. Now here we have another example. The example of P53. This is the entire P53, or this is, no, I can't remember. It's not the entire P53, that's a P53 domain. So this is a particular protein here. Yes, P53 human. It cannot be the entire thing though. I'm not entirely sure. But What this one here shows is the average over the 19 non-native substitutions over the residue number, from residue 1 to 385. Anything that jumps at you? This is the PFAM annotation. It's a domain or a functional unit. And I mean, the obvious, sometimes it's difficult, the job of being a teacher. It's so obvious. On the other hand, sometimes the obvious is not that obvious. So in this case, I assume that you all have seen the obvious. The point, obviously, is that the fact that over the course of this region, you find a signal in SNAP that correlates with the domain or with the unit is surprising. Functional impact has no relation to domain. Predicting for every single residue the effect of 19 non-native, that there would be any signal is not to be expected. And that this signal would at all, in the first case you try, correlate with something that is an annotation, is very surprising. Right? And it begins to tell you or show you that there may be a lot in this method that could become interesting. This one here talks about the prediction of hotspots, for which you can use it. In fact, it works to some extent. This is what this one says. This one talks about, in fact, the red that I showed, the degree to which you can see the binding site. Red is effect. Now you see different effects here, and the different reds, and ultimately that depends on how you compile effect. So, and we're getting back to the question that we discussed on Thursday. The question being, what do you expect if I take an average single domain plotting and I substitute every single residue by something else? What fraction of these mutations or variants will affect function? Most, or will most not? And ultimately here the level of red is in some sense moving that threshold. But it's a little bit more than that. This one here puts in the idea that what you may want to look at is when you look at, for instance, transmembrane helix. In that transmembrane helix, you know that hydrophobic residues can be exchanged against hydrophobic residues. Maybe you don't care about those. What you care about are the predictions for residues where you don't expect that they're neutral from the biophysics of the problem. And now let's assume that SNAP finds position where you assume that hydrophobic to hydrophobic, R to K, so charge to charge, would be neutral, and nevertheless SNAP predicts an effect. Those are interesting cases. And that is in fact this way of looking at it. And this way of looking at it really zooms completely into the binding site. Unfortunately, we have tried to make this work for a larger data set and still have so far not succeeded. This is an in silico mutagenesis for a large example, and I showed the LAC1 repressor from E. coli. That's the experimental data. Where for every single position they have over 4,000 mutations. For every single position they substituted over 10 particular amino acids at that native position. Black means effect, strength of effect, so most mutations make an effect. And somehow immediately just looking at it for a few seconds, you see that there is a clear correlation between predicted and observed. This is 5 years, this is 5 minutes. Well, now the method is available, this is 5 minutes. And it's remarkable how much information that captures. Here's a totally different story, the melanocortin receptor, where in fact Jana annotated for all the variants that we had that were predicted to have a strong effect. She annotated what is known experimentally about that function. That got us to some residues for which we had a pretty strong effect that were not studied experimentally, and that in fact led experimentalists to study those residues, and that led to interesting insights. Now here's another example of something that you can do with that method. And I should use the new slide because this is not clear. So what I'm looking at here are the melanocortin receptor for our human and another similar receptor. Overall, I cannot quite remember, I believe the level of sequence identity between these two proteins is in the ballpark of 60%. Overall, they're very similar. Now, what this method allows you to do, and from this slide even I have a difficulty to spot these examples. I believe those are the kinds of examples. We have here, I really should use my other, here, no. Here's an example. So what it allows you, these D, in this particular position, both proteins have a D. Overall 60% sequence identity, that particular position, they're identical. From the alignment, you could not at all predict that they would behave differently at that position. They both have a D. SNAP predicts a different effect of a mutation, of a susceptibility to mutation of these positions. Through that, it can predict at positions where you have the same residue aligned in an alignment. It can predict whether or not at that position where you have the same residue, they are similar. Essentially from asking, not is that a D, but in its context of the sequence, is this D as sensitive to change as that D? And if it's not, then it will behave differently. And we have a few examples where we can show that this in fact captures an important reality. There's no other tool in computational biology that gives you, or in bioinformatics, that gives you the opportunity to say something differential about the same residue in an alignment. Another great feature of this method. You can look now, instead of looking at the effect upon function, you can look at the effect on structure. And that is, in fact, I talked about that on Thursday. Regular secondary structure with the stance mutation or variant, disorder does not. And that is, the work is done by Chris Schaefer. And this is the final result. Content of helix is measured here, length of a helix. This is pairwise sequence identity. So we look at a protein, we mutate a few of these residues, we keep mutating until we have some random overlap between the two. The different colors are different ways of mutation. So at every single time point, you change a residue, or you decide whether you change, and that changes according to some probability. Those are different probabilities. Overall, you see that the content of helix, predicted helix, stays constant. The content of strand stays constant. The average length of a helix essentially stays constant. The average length of a strand stays constant. That's surprising. So you begin with a protein, you mutate to something that has only 5% sequence identity to what you had originally, and you do not choose. This has nothing to do with evolution. You do it in silico. So completely change the sequence, and overall, over the data set that we looked at, I can't remember 1,000 proteins, this stays the same. That was absolutely unexpected for us. Alluded to that on Thursday as well, when that's sequence running from residue 1 to, in this particular case, to a residue 149, red helices, beta strands in green. So that is the sequence at one particular time point. That is at another time point, another time point, another. The more we mutate, the more we go down. So sometimes you see that you begin with a beta strand and you mutate into something that is predicted to be an alpha helix. Sometimes you begin in a region, it's the same here. Sometimes you begin with an alpha helix and you mutate into a strand. So you mutate strands and helices into each other. And you see that quite often, actually. This is just 4 examples that we looked at. Everything we looked at looked somehow like that, which was surprising to us. Now, the disorder story, background again, we don't know quite what disorder is. One definition is you shine light at 2 different time points, you don't see the same thing. And when we look at disorder, long disorder, so now we look at disorder that is at least 30 consecutive residues, then for that we see, in fact, the content in disorder clearly goes down. Wait a minute, what is this one now? This one doesn't. Content in disorder. Why What is this? I don't know what this is. I would have to look up the paper. So this is, this doesn't exist, because I don't remember what it is. The point here is alpha helices, beta strands remain the same, are relatively resistant to mutations. Disorder is not, because it's highly non-trivial domain disorder. Clearly goes down the line much more than the helix, and I should put that one to the other. If so, then we should be able to infect. No, not if so. That is one reality. Another reality is, again, I said that we don't have enough data to predict the effect upon function. We need better data. Why not use structure? Why not predict the effect upon structure? If we knew the effect upon structure, we could actually also distinguish in a mechanistic model. One method predicts the effect upon function, one method predicts the effect upon structure, and that would help us to sort of get the whole story together. That's why we began working on structure. Working on structure, we actually string up the entire PDB in one big sort of mega protein in some sense. That's the way Chris begins. And then we overlap 5 mers. And the reason for choosing 5 mers has a longer story in itself. And we wanted to predict the difference between an example where essentially here the mutation changes the structure and where it doesn't change the structure in the 5 mer. So here are 2 examples. So we want to distinguish this case from that case. And to some extent we can do that. Let's not get into the distribution. That is the way the method predicts to some extent the ability to predict structure. Now, does the method make a difference? Does it work? And one way to answer that is now you want to apply it and see whether it predicts some signals of function. And the signals of function that we look at here is stability. So that is the delta delta G. We have a large database of PMD where we see the effect upon the binding energy of a mutation. And function here is the data that we use for SNAP. The method that we now use has been trained only to predict the effect on structure on 5 mers. So we take the entire PDB, we align everything against everything, and we look at 5 mers that change according to some criteria on the 3D structure and those that don't. Okay? And now we have 2 data sets. In dark gray, no observed effect. Light gray observed effect. Does our method capture the reality? Right? It's complicated. It's very, very complicated because there's so much overlap. There's no, it's the example where you cannot really find a threshold that is safe. You have almost entirely overlapping distributions. Now, what do you use that for? You could say, well, my method totally failed because here I have a completely new data set and it cannot answer that. On the other hand, you develop the method to distinguish the effect upon structure. None of those is the effect upon structure. That's the effect upon function or some other criterion, right? So you rightfully can ask, well, I was not really supposed to be able to do that. So what other tests should I try? Do you have an idea what we could try? So this is one of those non-happy end stories in this particular case. We did not find the test case that made the developer, Chris Schaefer in this particular case, happy to go on. Chris gave up. So this is an example where it got the publication, it got the PhD, we have a method that works, but nobody uses it because the developer was not impressed by this graph. In some sense, this graph was for Chris and for that method the end of the line. And I'm not showing it for you to learn anything. Well, actually I do. For me, this is not the end of the line. This cannot be the end of the line. This is too unclear. Maybe that brings us back to the exercises. Nail as early as possible. Pursue when you have a suspicion. Almost everybody in the room I've heard to make a statement and not really get down to the heart of it. Be more pushy when you have an idea. Formulate ideas, hypotheses about your data early on for whatever you look at. Time is short, and push. Then push for it. And then follow it through to the end. Anyway, so we have the method, and we still don't quite know to the extent it works. I am, on this particular case, more optimistic than the developer, but nobody really uses it. Here's another idea, and that is the MD idea coming originally from Mark Hoffman, and that idea was, let's just look at how variants would behave if I did an MD simulation. So I'm looking at two variants. I'm asking, MD you can only apply when you know the three-dimensional structure. So essentially, what's the effect of that mutation or the variant upon the dynamics of a structure? And the task that originally was set to Mark was to in fact see whether for some subset for which we know three-dimensional structures, this kind of idea could give us new reality as opposed to SNAP. Could that do more than just tell us something has an effect or not? Right? For some examples, and I will show you two here, this worked. Here is one, the Gaucher disease, where the dynamics showed that a particular variant, why a particular variant was important. Not only that, actually it also suggested how you could recover on the disease-causing variant and what you possibly could do in order to put a drug that would help. That is work that he started in Israel, in the Futterman group. And here is a piece that he did in collaboration with Thomas Meitinger and Tim Strom, rechts der Isar, where in fact they had data for Parkinson patients, and they had a lot of data, and some of these data for them in terms of statistical significance stood out from looking at the sequencing of families, and in fact stood out in the sense those were, they believed, were mutations that were the best candidates for being relevant for Parkinson. And by the molecular dynamics simulations from Mark, we got the same evidence for what stood out in their statistical sense, and we looked at it from our view, and these two things collided and gave a nice publication.